Enzyme catalysis. Now, after understanding the basic types of the catalyst, the process catalysis, the mechanism of the heterogeneous catalysis, and then doing shape selective catalyst zeolites. Now, let us move to the next topic, which is called enzyme catalysis. See, enzymes are basically complex nitrogenous molecules, organic molecules, which are having very high molecular masses. Chemically, they are protein in nature, which are having very high molecular masses. In nature, they exist in colloidal state in solutions. Now, we know these enzymes are the ones which are responsible for catalyzing the millions and millions of the reactions which takes place in plants and animals in our body. So that is why what we call them as biochemical catalysts. Remember, enzymes are called biochemical catalysts, and this process or phenomenon is called biochemical catalysis. The reason is, you need to understand this. We have millions and millions of reactions which are taking place in our body and in the bodies of the plants and animals. All these reactions are catalyzed by these nitrogenous complex organic molecules which are proteinous in nature. Now, that is why these proteinous in nature molecules, which are nitrogenous in nature, having nitrogen as the main constituents, are called biochemical catalysts because they are catalyzing biochemical reactions, and this phenomenon is called biochemical catalysis. Now, we have millions of examples. We can take the most common which we will have been studying from the junior levels are to understand these biochemical catalysts are. We know, like if we take sucrose C12H22O11 in water and we put enzyme called invertase, what we get is C6H12O6 plus C6H12O6. So it is glucose and fructose. So your normal sucrose is broken down into glucose and fructose by the use of an enzyme called invertase. Remember, we put ase as a suffix to the names of the enzymes. Now then this glucose C6H12O6 is converted into alcohol, 2 molecules of ethanol, and releases water. This glucose is converted into alcohol by an enzyme called zymase. So invertase converts sucrose to glucose and fructose, and zymase converts glucose into alcohol and water. Now we have seen that the zymase converts the glucose into the 2 molecules of alcohol, which is coming out in the form of an aqueous medium. But remember, along with this, 2 molecules of CO2 also comes out. So this is how the equation is balanced, and it is basically the zymase is the enzyme which is converting your glucose into 2 molecules of alcohol and CO2. Now let us see some other examples. Let us see the example of the starch C6H10O5. We know it's basically is a polymer of the simple glucose molecules, which is given as C6H10O5. So it is this is your starch, which when combines with water in the presence of an enzyme called diastase is converted into C12H22O11, is broken down. So this starch is broken down into your normal sucrose molecule. These are which is actually is a disaccharide. Now the moment it gets converted into this one, then the C12H22O11, on this what happens is the enzyme called in the presence of again this is taken in the aqueous state, the enzyme called maltase convert them into C6H12O6, which is maltose. So these are two enzymes, diastase and maltase, which converts the starch into maltose. So it is very similar to what we have done, the invertase and the zymase. Let us take some other examples, like we know a urea molecule, which is NH2CONH2. Now this molecule in presence of water is converted into ammonia and CO2 by the simple enzyme which is called urease. In a very similar way, we know we have pepsin, which is secreted in our stomach along with the HCl to break down the food substances, basically acting on the protein molecules. We also know how the milk is converted into curd by the enzymatic activity. So we know a number of examples where we find that they basically these biochemical catalysts are responsible for increasing the rate of these normal biochemical reactions, and they play a very active and very important role in these biochemical reactions. Now let us study the main characteristic features of these enzymes.